to our Chapter 11 discussion on Mendelian genetics. I think the end of this year with genetics is some of the most exciting part of biology, so I'm really excited to get into it with you. So it's important to think back to our previous discussion on the cell cycle and especially meiosis. The behavior of chromosomes during meiosis that makes those gametes is what allows for heritability. Events like crossing over and independent assortment in that first stages and that first division of meiosis allow for the patterns we're going to see in inheritance. If you think about those homologous pairs of chromosomes, you have a 50% chance of inheriting one or the other of these homologous pair. That means if you look at a single allele on a single gene, you have a 50% chance of getting one allele versus the other. And these 50% ratios are going to make for nice and easy to use ratios when we look at inheritance probabilities and we start playing with some Punnett squares. So we need to go back in time, though, to see how we figured this all out. And it all goes back to a monk living in what is now the Czech Republic, whose name was Gregor Mendel, known as the father of genetics. And he determined how to analyze these genetic patterns from a scientific and mathematical perspective. And he was doing this in the mid-1800s. In fact, his research was actually published around the same time as Darwin's publications, but Darwin never read Mendel. He was sent Mendel's work, but never really read it, and it was very much considered kind of that missing piece to his understanding of evolution and how these traits are inherited. So what Mendel did was he established true breeding lines for particular traits. So that means that both alleles are the same on either side of their chromosomes. And then he started crossing these lines, so mixing these traits, and then he would just count the offspring and see what happened. And he started to notice really clear patterns. And in Mendel's data, he followed 7 different easily identifiable traits. He looked at seed shape and color, he looked at flower color, position, the height of the plants, the pea pod shape, and pea pod color. And all of these were just easy to observe, didn't require any scientific equipment beyond just the untrained eye. And what he did was each cross, he would see how many offspring of each trait arose and then continue to cross them to see what patterns he observed. So let's take a look at his data for just the seed shape and color. So we're going to have 3 different generations we look at. The P generation is the parental generation. These are our pure breeding lines. The F1 is the first generation from the parents, so that's the progeny or the offspring of the 2 P generation plants. And then the F2 is the second generation, and you can imagine it continues there on F3, F4, F5, the progeny of the previous generation. And so we're going to look at seed color, yellow and green. He started with a plant that had yellow seeds and a plant that had green seeds. Same thing, he did that with round and wrinkled seeds. And so the P generation, we have an equal number of yellow and green, round and wrinkled. But then he crosses them in that F1 generation. 3,841 of the offspring plants, he was doing big plots, were all yellow. And same thing, 4,023 of his offspring in this generation were all round. So then he crosses those again, plants these fields, and he finds that he, in the F2 generation, so he just crosses only F1s together, he gets 6,000 yellow seeds to 2,000 green seeds. Again, a very similar ratio, 5,474 round seeds to 1,800 wrinkled seeds. So what do we start to notice? What ratios do we see here? Well, we have kind of a 1-to-1, a 1-to-0, and then if we divide this out, 6,000 by 2,000, pretty darn close to 3-to-1 ratio. Same thing here, 5,400 to 1,800. It's basically, if we divide that out, 2.96 to 1, really close to 3-to-1. So we're starting to see in these second generations from those pure breeding lines a 3-to-1 ratio of offspring. Three of these offspring have one trait and one has the different trait. And that different trait is always the one that disappeared in that F1 generation. So these ratios are going to be really important as we start to look at genetics. So let's put some vocabulary and some of these conclusions together. So for any given trait, Mendel found that when you crossed two different lines, the first generation always showed just one trait. There was never a mixture. It was always just one trait that was shown. Then when he crossed those F1 generations, both traits were shown, but it was always in roughly a 3-to-1 ratio. So it didn't matter if he was looking at flower color, height, pod color, flower position, it always appeared in that F2 generation in that 3-to-1 ratio. And so we start to get some of these terms. We've already talked about how a gene is a unit of DNA that codes for a protein. And a trait is what is the expressed form or what can be seen. So like in that previous example, flower color or seed wrinkliness. And then an allele is the alternate form of a gene, which allows for different traits from the same gene. And we're going to express these alleles with capital letters or lowercase letters. And there should always be two letters. So big A, little a, or little a, for example. But it's important to remember that these alternate forms of a gene mean that there's going to be some variations in the genetic sequence. That's what's causing these differences. Overall, it's almost exactly identical with maybe just a couple mutations that make it different, and that's going to affect how the protein changes. So for example, the allele for purple flowers codes for an enzyme that causes that purple coloration. It makes that purple pigment. The allele for white flowers, it's possible that that sequence no longer codes for that gene or that enzyme. And so that enzyme isn't made. And so then there's no purple pigment made. And if there's no purple pigment made, that flower is going to appear white because it's the absence of any of those pigments. And so this is how you can see how this actually plays out in reality. So we can see that if I had two alleles for purple flowers, I'm making a lot of enzyme. I'm going to be very purple. If I have two alleles for white, I'm making no enzyme, and I'm going to have white flowers. If I have purple and white, I still have some enzyme. I may not have as much, but I'm still making purple pigment, and that's going to overwhelm the white petals. So it's important when we talk about this, we understand the difference in some of this vocabulary. So you're going to hear the words phenotype and genotype a lot. Phenotype is the trait that is actually expressed. So this is using your words. So I always remember this because the phenotype describes the physical trait. With genotype, we're talking about what are the alleles? What's that actual genetic makeup? So here's where we're using those letters. So like big A, or big A, little a. And I remember this because the genotype is what is coded for in the genes. So what's actually in the genes versus the physical trait. And not all alleles are the same. So like we saw with that flower making the enzyme for pigment versus not making it, some are going to be dominant because if the recessive trait just doesn't code for anything, even if you only have half the amount of enzyme, you're still making something, and that's going to overwhelm that white flower. So we're going to use the capital letter A, or any letter that is, for dominant, and it is always expressed if it's present because it's making that enzyme that's overriding the recessive allele. The recessive allele will use the lowercase letter, and that trait is only expressed if both alleles are recessive. So you have to have little a, or whatever letters you're using, and it's always going to lose out to the dominant trait if it's mixed. So going back here, we can see, even though we have one allele for white flowers that's not making any enzyme, because we still have that purple making enzyme, the dominant trait will win out. And so this is a great visualization of why dominance wins out, because we're still producing an enzyme that's still making purple. And so even though half our genes are coding for no enzyme, we still are making some of that purple pigment, and the recessive can't really fight it. So two more words I want you to be familiar with. Homos is Greek for the same. Heteros is Greek for the other of two, or different. So we're going to be referring to these allele combinations as homozygous if they're the same. So like dominant or recessive, those would be homozygous dominant or homozygous recessive. If there's a mixture of alleles, a dominant and a recessive allele, we're going to call that heterozygous, because we have both, or the different ones that are present. And so just when we look at this, our genotypes, when we're homozygous, will either be homozygous dominant, homozygous recessive. We could refer to that individual as a homozygote. And heterozygous is big A, little a, which will express the dominant phenotype. So remember, the phenotype of someone who's heterozygous will be no different than someone who's homozygous dominant, but their genotype is very different, and that'll come into play when we start looking at Punnett squares. So we could refer to that individual as a heterozygote. And it's important to remember that heterozygotes are just allele uneven. Bio memes never get old. Okay, so in summary, genotype are the alleles the organism has. Phenotype is the trait that's actually shown. And remember that dominant wins out because of some genetic quirk. Maybe it's producing something, maybe it's making more of something, but it's going to be overly expressed versus the other one. And that's just how the proteins end up working. So some quick practice here. We have a fluffy little cat. And in cats, long hair, shown by the allele lowercase l, is recessive to the dominant trait of short hair. So short hair is the dominant trait, which would be shown by the allele big L. I want you to look at this cat and tell me, what is the phenotype and the genotype of this cat? I'll give you 5 seconds. Well, if you didn't pause, here we go. Phenotype is long hair. If you've never seen a cat before and you don't know that this is long hair, this is a pretty long-haired cat. This is a pretty bushy-looking cat. And so its genotype, because we know that long hair is recessive, has to be little l, or homozygous recessive. Another term I want you to know that you can use for those heterozygotes is a carrier. Carriers possess the allele for the recessive trait, but it does not actually express it. When neither parental flower is white, but offspring flowers are white, that means the parents were carriers. So here you can see no parents have white flowers, but they produce white flowered offspring. That means that these parents are carriers for the white trait. And both parents must give their offspring the chromosome with the recessive allele. So if they're heterozygotes, they both have to give that recessive allele through the process of meiosis. And that's going to be the foundation for what builds our Punnett squares. So how do we figure out what possible genotypes and phenotypes there could be? By using a Punnett square. And when we're analyzing a single gene like this, we're going to call it a monohybrid cross. And I'm sure those of you who are thinking mono means one, yes, there's the possibility for dihybrids or trihybrids or pentahybrids if we really wanted to go so wild. And basically, how it works is it's just a little graphic organizer that's going to let us predict the outcomes of meiosis. And then what could be the outcomes of those mixing of genes through random fertilization. So let's say our father is heterozygous and our mother is homozygous recessive. Doesn't matter if you put the father on top or on the side, mother on the top of the side. Doesn't matter where you put them. But you're going to just put one allele from each gene. Because remember, the sperm and the eggs are going to be haploid. They're not going to have pairs of each chromosome. They're just going to have one of each chromosome. So that means each gamete or each sperm from dad is only going to have a big B or a little b because they're not going to have both chromosomes. Mom, it's either going to have one little b or one little b because she's homozygous recessive. She's sending the same either way. So what you do is you drop it down and put it over. So whatever's above it and to the side, just fill that into the box. So a big B from dad, a little b from mom, a little b from dad, a little b from mom, a big B from dad, a little b from mom, a little b from dad, a little b from mom. Now, typically when you're writing this, you always put, even if mom had the big B, it doesn't matter. The big B always goes first. Your dominant allele will always go first. You never want to see like little b, big B written down. You can, but it just is going to complicate things. And so make sure you do that. And so if this gamete if this sperm mixed with this egg, this is what we get. If this sperm mixed with this egg, this is what we would get in the offspring. And so it helps us predict, because we don't know which one's going to be used because of random fertilization, we can predict the ratios. So we can expect that if these parents were to have children, there's a 50% chance, because it's 2 divided by 4, that they'll be heterozygous, and there's a 50% chance that their offspring will be homozygous recessive. And this is all because of the law of segregation. Remember that during metaphase and anaphase 1 of meiosis, each gamete is only going to receive one allele for any trait. They're going to split the homologs. So organisms produce an equal number of gametes with each allele, because one gamete gets the one gamete gets the other. So there'll be an even distribution or even segregation of those alleles. And so we can start to predict what offspring are going to look like. It's a good note to say that I hope none of you say that this is how panda bears are actually made. It's not by mixing a polar bear or a black bear, but it does give us a good example of like how this can work for some traits. So when we look back at Mendel's P's, what are the dominant traits? Well, we can see that when we mixed yellow and green and all of them were yellow, or our heterozygous F1 generation was all yellow, we can assume yellow is dominant. Same with round. Because all the heterozygotes were round, we can assume that that is our dominant trait. And if we were to do a Punnett square, we would find very similar ratios. So that process of independent assortment, which means that each pair of homologs can line up differently, means that different genes are not linked together. If they're on different chromosomes, inheriting one doesn't mean you also inherit another. And this is what builds the foundation for making bigger Punnett squares, is we can take those possible combinations of alleles and put them on either side, and we will get all possible offspring. And this is because those proportional numbers of all gametes with all those different combinations are produced through meiosis. And so we sometimes will get offspring that have totally unique traits from their parents, just totally different combinations of traits that were visible in either parent. So for example, we may find a green round pea that neither parent was green or round because there were yellow round parents, or yellow wrinkled peas that weren't visible in either parent. And this is going to be an example of what we're going to call recombination. So let's talk about crossing multiple genes at once. And you can see in this diagram that if we are mixing two genes here, so like round and yellow, we can take different one allele from each gene there, and we'll end up with 2 alleles, and then we'll combine those and we'll end up with, again, 4 alleles for 2 chromosomes, and we can start to predict ratios for bigger numbers. So we can run a Punnett square for both at the same exact time. So this is going to be called a dihybrid cross. So we're going to be crossing 2 plants from the F1 generation to make the F2 generation, and we're going to observe seed color and seed shape. So what are the genotypes of our parents if they're going to be heterozygous for both? Well, that's going to be big R, big Y, little y. Now it's important to note, you're never going to write RY, RY. You're always going to keep your R's together and your Y's together. R's are going to be together and Y's are going to be together. And that's going to be really important when you start doing your Punnett square. You're going to be tempted to do like big R, big Y, little r, little y. No, keep your R's together, keep your Y's together, just like you always keep your dominant or capital letters first. So what are all the possible gamete combinations that we could make out of this through independent assortment? Well, let's start figuring it out. So if we took the big R and the big Y, we would have one gamete that's big R, big Y. And remember, this is on one chromosome and this is on the other. So you have a 50% chance, each gamete has a 50% chance of this allele or this allele. At the same time, it also has a 50% chance of this allele and this allele. So we end up there. Since we have a 50-50 shot, you could get a big R and a little y together as a gamete. You could get a little r with a big Y together. And you could get a little r with a little y. Remember, all of these possible gametes are possible because of independent assortment in meiosis. Meiosis is essential here. So let's fill in those gametes that we have in our Punnett square. So we said we have big R, big Y. Remember, big R, big Y, big R, little y, little r, big Y, R, Y. And because mom is the same, we can put mom on the side here. Now, we're going to start filling this out. So, same thing we did in the monohybrid cross. We drop down and slide over. So, when we drop down and slide over, we see, I should have left the, there we go. We see big R, big R, big Y, big Y. Now, this is where I said it's very tempting to write RY, RY, because you drop those down together. Keep your R's together, keep your Y's together. And so as you fill that out, you're going to see, and we have RR, RYY, big R, little r, big Y, and so on. All these different combinations. And so now I'm going to ask you a question. What is the probability that the offspring would be both green and round? Well, there's 16 total boxes here. And I'm looking for green, which is little y, and round, which is containing a big R. So we have 1, 2, any others that you see? 3, very good. So there's 3 instances where we have little y, and at least one big R to make it round. So that's 3 divided by 16, or a 0.18 or 18.75%. So that's the probability, is 0.18 or 18.75%. Now, is there a shortcut to all this? Because I'm sure you're imagining, I don't want to do that now with 3 or 5 or 6, or even that takes a long time. Well, let's think about statistics here. So the inheritance of unlinked genes and independent events. Statistics tells us that the probability of A or B happening is the probability of A plus the probability of B. If we want to know the probability of A and B, it's the probability of A times the probability of B. So if we figure out the probability that it's round and the probability that it's green, we can just multiply those. So let's see how that works. Probability that it's round. If I do a monohybrid, that's big R, little r 1, big R 2, big R, little r 3, little r, nope. So 3 out of 4. How many will be green? Well, only one little y when I did the cross here. And remember, these are the same alleles I got from the parents. That's 1 out of 4. So if I do 3 out of 4 times 1 out of 4, that is going to be 3 over 16. Or if I do the percentages, 0.75 times 0.25 is 0.1875, 18%. The same thing. You have a shortcut. If you just do the monohybrid crosses and multiply, you'll get the same answer. It's going to take you a lot less time. Because what if we did a trihybrid? You do not want to fill out a grid this large. It's going to take you all day. So use that cheat code. Instead, do probability of A times probability of B times probability of C. Figure out your answer. Same thing here. You don't want to mess around with a pentahybrid cross. So do your shortcut. Now, there's another type of cross called a test cross. And this is, say you find a flower and you don't know, is it dominant? Is it recessive? Is it, heterozygous? Or, sorry, is it dominant or heterozygous? Is it homozygous, dominant, or heterozygous? And what you're going to do is you're going to breed that unknown phenotype, because remember, if you see that dominant phenotype, it could be homozygous dominant, it could be heterozygous. We don't know. So if you breed that with a recessive phenotype, which is a known phenotype, like with these flowers, you know the white flower has to be little p, but a purple flower could be one or the other. Well, breed them together. If the offspring are all purple, you know that original flower was homozygous dominant. But if you get some white flowers, you know it had to have been recessive, because the only way you can get recessive is if you get a recessive allele from both mom and dad. So, I have a little question for you. Do you think test crosses are good ideas for testing humans? So if you don't know if you are homozygous dominant or heterozygous for a trait, do you think you could do a test cross to really figure that out effectively? Seems like it'd take a while. Last thing I want to talk about is the concept of epistasis, and this is going to be something that where the presence of one gene can mask other genes in the pathway. So sort of like when we were looking at those proteins and the recessive form doesn't produce it, this is where a different trait can kind of override another gene. An example of this is mouse fur color. The allele for agouti coloration, this nice brown coloration, is dominant, or so we'll use big A, for the allele for black coloration. We'll use little a. And so you can see that there's going to be more agouti than black. However, there's another gene where if you get the homozygous recessive form, causes albinism. And it doesn't matter if you have the traits for agouti or if you have the traits for black fur. If you have the trait for albinism, it's going to overcome everything. So here we can see a third type of mouse present because this is going to override. And so this is called epistasis, and it's not extremely common, but you will find that there's all sorts of different genes that play off one another, and presence of one can completely mess with the presence of another. And that's why you may be looking at like your hair color and your families and be like, I don't get if this is dominant or recessive. Well, sometimes many genes are playing off at one another, and we're going to really dive into that in the next lecture with non-Mendelian genetics. So last thing I just want to quick cover is pedigrees. I'm going to move my face here so you can see the notes there. A pedigree is a graphic organizer that's going to show how a gene is passed down through generations. And we're going to do this in class together. But basically, blank shapes are those without a trait. If it's filled in, you have the trait. So whether you're looking at, this is not, and this is really important, this is not dominant or recessive. It's not filled in as dominant, blank as recessive. You're looking at a particular trait. So maybe you're looking at a recessive trait. That's going to be filled in. Maybe you're looking at a dominant trait. That's going to be filled in. So you're just looking at one phenotype. What is that phenotype that you're following? If you half shaded in, that means it's a carrier. Remember, if it's a carrier, you're going to be following a recessive trait, because if it's a dominant trait, any carriers we know are just expressing that trait. But we want to know who the carriers are when a recessive trait, because it helps us figure out what the offspring could be. If you see two together like cherries, that means they're twins, but otherwise squares are males, circles are females. When we're counting it, just know that you want to count both generations and individuals. So generations are going to be Roman numerals, and individuals in each generation are Arabic numerals, and we start over in each generation. And this is going to help us find people in the pedigree, because we don't want to have to worry about scribbling in names everywhere. So it's much easier to say, oh, person 37. Yep, I can find that really quickly, instead of being like Jennifer. Okay, where is Jennifer? I don't know. So always number generations with Roman numerals, Arabic numerals for individuals, and restart your numbering with each new generation. Typically, you're going to have older people to the left, younger people to the right, but because of later generations, it gets harder to do that. So try, but it's not essential. Connections on our pedigree. Well, this means that they had kids together if they're connected by a horizontal line. If they have branching lines above them, that means they're siblings. So you can see that they had kids together. They had 3 kids. They had a boy and 2 girls. One of the girls had the trait. Now, every year kids always do this. They draw it down and connection, trying to draw siblings. This is not siblings. This is not a couple who had 2 sons and a daughter. This pedigree shows a couple who had 1 daughter, and she's doing really well for herself. She has 2 husbands. She's doing great. But this doesn't show 3 siblings. This shows 1 child from these parents and her 2 husbands. And then especially avoid closed loops. This pedigree, anytime you show a closed loop, what you're showing is that 1 couple had 3 children, 2 sons and a daughter, and they're all intermarried. Don't, when you do your pedigree, make a closed loop. That's a problem. So when we're looking at pedigrees, a couple things that'll help you find whether it's dominant or recessive. In a dominant trait pedigree, every affected individual has at least 1 affected parent. So if you have it, at least one of your parents has it because they had to pass along that trait. Affected parents can have children who are unaffected. So here we see like this person and this person both have it, but they have a kid who doesn't. It's because they're both heterozygous, and so they both passed on the recessive trait to person 3-2 down here. In the recessive trait pedigree, you can skip generations. So an individual who is affected may have parents who are not affected. So here we can see neither person 1-1 or 1-2 has the trait. They are carriers for it. And then they have a child, person 2-1, who does have the trait. And so that means is that these people are both carriers because they could pass it along, but they don't actually have it themselves. All children of 2 affected individuals will have to be affected. So if you see 2 people who are affected and they have a child who is unaffected, can't be recessive, because remember, if you're affected, you only have that little allele, and little alleles are only going to make more little alleles. And then you can draw in carriers. It's important to note on a question, you may not see carriers because it's asking you, is it dominant or recessive trait we're looking at? And so the carriers are kind of a dead giveaway, so they don't always draw in carriers. You should draw in carriers if you're asked to draw a pedigree, but if a question shows a pedigree and there's no carriers, don't automatically assume it's dominant. Look at some of these other clues like skipping generations. That's all I have for you on this. Some couple of take-home points I really want to make sure you understand. I covered it 1,000 times, so please make sure you understand the difference of dominant and recessive, why things can be dominant versus recessive with what the protein is made and what its job is. Make sure you understand the difference between heterozygous and homozygous. Make sure that you understand how meiosis is involved, the law of segregation and independent assortment, and how that lets us predict things with Punnett squares. Make sure you understand the differences between a dominant and a recessive pedigree and be able to draw and interpret a pedigree. And finally, be able to do monohybrid, dihybrid, trihybrid cross, because you never know what's going to get thrown at you. All right, have a good one, and hope you learned something.